Good morning, dear students. Today we are going to start a new topic that is mitochondria. I hope that you all are familiar with mitochondria, what is its importance. Anyhow, without wasting time, we can move to our topic mitochondria and chloroplast. Both these organelles are relatives, both are energy converting organelles and both are found only in eukaryotic cells. Okay, so this mitochondria is present in animal cells while chloroplast is present in plant cell. So that is the only existing difference between these two organelles. And all the other respect regarding with regard to the function, they are same. And this mitochondria is known as powerhouse of the cell. Why it is known as the powerhouse of a cell? Because this mitochondria, it is tapping the energy that is releasing out of the metabolism of foodstuff and convert this energy to another energy rich molecule like our boost or horlicks, that is ATP. So it is producing ATP. So that is why mitochondria is known as the powerhouse of the cell. And we need to be familiar with the names of some scientists that are pegged, means that are linked with mitochondria. So while whoever is learning about mitochondria, they all should have to hear about the names of all these scientists. So just we are moving through each name of this scientist one by one. Kolliker, very important person. He found the mitochondria in 1857. So not that name, Kolliker. Then Benda, he named. So whenever a baby is born, then need to give a name. So that name for our baby, it's mitochondria. Yeah, its name is given by Benda, 1898. Then another scientist, Kaelin and Kalkar and Belitzer, and what their importance, they studied electron transfer system. What is this system? We will learn later in this chapter. Just hear that process, electron transfer system and oxidative phosphorylation. These scientists studied about these two very important processes that are the key steps that ultimately results in the production of ATP inside the cell. Then Claude. Claude is another scientist in 1940. He isolate mitochondria from where? From liver cells. Next, another two scientists, Kennedy and Leniger in 1948. What their contribution? They proved. What they proved? They proved that Krebs cycle and oxidative phosphorylation both are also very important metabolic pathway that is happening inside the mitochondria. There are also some steps towards the synthesis of ATP. Anyhow, we will learn about these two processes also later in this chapter. Now leave it. Just learn that this process is happening inside the mitochondria. It is proved by Kennedy and Leniger. I know that it's very difficult to memorize the names of all the scientists. Anyhow, you should be familiar. Just have a feel that I heard about these names like that. Then Peter Michel et al. What his role? Peter Michel et al. Means his associates in 1961. They elucidate the mechanism of oxidative phosphorylation, chemiosmotic mechanism. No need to note that specification. Chemiosmotic, that too we will learn later in this chapter. Chemiosmotic mechanism of oxidative phosphorylation. That is his contribution. Okay. Then for that finding, he received Nobel Prize too in 1978. Then another two scientists, Paul Boyer and John Walker, 1997. and they described how the ATP synthase, it is a final motor inside the mitochondria that is synthesizing the ATP. So how it function, that mechanism is explained by Paul Boyer and John Walker in 1997. I think these are the some very important scientists who brings about findings associated with the mitochondria. Then we are moving to another important section, that is endosymbiont hypothesis, and this is asked so many times for the university examination. It's a very simple concept. Anyhow, I am describing you. Somehow you might have learned about this hypothesis in your lower classes. Anyhow, um, what is this? Is a bacteria cell. Bacteria cell, and this chloroplast and mitochondria, they once billions of years back, they exist inside the bacterial cells. They are the organelles inside bacterial cells or a prokaryotic cell. So we can see that that mitochondria, chloroplast also present inside this type of prokaryotic cells billions of years back, and then it somehow get inside, or somehow it this eukaryotic cell engulf that mitochondria and chloroplast around billions of years back. And after it enters inside the body of eukaryotic cell, it um, brings about an association, a relationship. What type of relationship? Symbiotic relationship. They started a symbiotic relationship association with this eukaryotic cell. And what type of association that between that mitochondria and the eukaryotic cell? So let's see. This mitochondria and the chloroplast both have their own genome. They have their own genome. They don't need to depend anybody in anything. But after some years, they lose a few of its genome, and they have to depend on this eukaryotic cell. They have to depend on the proteins that is synthesized by the eukaryotic cell. Okay, so it is depending this eukaryotic cell, and what in turn this eukaryotic cell is doing, this eukaryotic cell depend on this mitochondria and chloroplast for their majority of ATP requirement. So that is the association, that is the symbiotic relationship. This prokaryotic engulfed prokaryotic cell is depending the eukaryotic cell for some of the proteins, and this eukaryotic cell is depending the prokaryotic cell for majority of its ATP requirement. Okay, so that is the symbiotic association. I hope that now that idea concept is clear to you. So that is the hypothesis. The hypothesis says that the energy converting organelles, we know that which are those organelles, mitochondria and chloroplast. That organelles are evolved from some prokaryotes by chance of engulfing, and after being engulfed, it established symbiotic relationship. So that is the hypothesis. Okay, clear? And one more thing we need to understand, that in the course of the time, this mitochondria and chloroplast, they lost, I already mentioned you that they lost some of their genes. And those genes, they are moving from them to the nuclear DNA. So, that nuclear DNA got some genes from mitochondria and chloroplast. And on reaching this nucleus, from that genes also, some proteins are forming. Some proteins are forming. These genes As later, after so many years, as the scientists examined, they found that these genes they resemble that of bacterial genes. So that's also a proof that this mitochondria and chloroplast they emerge from bacterial cells. Clear? So that's what it's telling that some of the genes present in the nuclear genome it resembles that of the bacterial genes. How it is possible? Is it clear? It's possible only by the way that these genes they are moved from mitochondria or chloroplast to the nuclear genome or nuclear DNA. So that's also a proof that mitochondria and chloroplast emerge. It is received by the eukaryotic cell from some prokaryotic cell. So that is endosymbiont hypothesis. Endosymbiont hypothesis. Okay.
I'm an icon, man. And my girl about a 10. I should fuck her in the Benz.